And so that tends to be fairly thin, fairly flexible. Outside of the sort of yeah, let's see here. So here's the primary cell walls here. After the plant has grown, it's a sort of a more mature plant. The cells are not the plant itself is not getting larger. Then what happens is that there's deposition of additional material, a lot of cellulose and some other components as well that create, at least in some plants, quite a thick secondary cell wall that's shown right here. So you can see that's really a much thicker layer than what you see here for the primary cell wall. And then in between, so these are two plant cells that are coming together. And in between is something called the middle lamella, which is a layer between the primary walls of adjacent cells. Okay, and so down here in this more schematic drawing, you can sort of see how these guys are actually organized with respect to each other. Here's the very thick secondary cell wall, the thinner primary wall that's right on the interior there, and then in between are these middle lamellar areas right here. And then very importantly, these cells are not completely isolated from each other. They're actually connected up with channels. And in plant cells, the channels are called plasmodesmata, and you see the connections right here. So these are actual links that connect up the cytoplasm of one cell. They cross the secondary and primary cell wall in this middle lamellar area as well, and connect to the cytoplasm of an adjacent cell. And so this is also super important, right? Because if these cells were really isolated from each other, they'd sort of be all potentially operating individually, right, doing their own thing. But in an organism, in a plant or a liver, a tissue, or other kinds of either tissues or whole organisms, the cells actually have to be cooperating with each other. They have to be exchanging information, and information in the cellular world is typically exchanged through molecules. And so these kinds of channels, and we'll look also at the types of channels that connect up animal cells, allow cells in a multicellular organism to interact with each other and exchange molecules. Okay, now I want to just talk a little bit about the extracellular matrix of animal cells. So you know that animal cells lack cell walls, but it turns out that they're actually covered by a pretty extensive network of molecules that are outside of the cell. And these include things, typically they're made up of molecules called glycoproteins, or in some cases proteoglycans. Now when you hear glyco, you know that has something to do with sugars, right? And when you hear proteo, you know that has something to do with proteins. And so typically if we call something a glycoprotein, we're referring to a molecule that is largely made up of protein, polypeptide, but has some sugar molecules attached to it. If we turn that around and talk about molecules that are proteoglycans, then we're talking about molecules that have a small protein core, sort of a central molecule that's made up of a polypeptide, that has a lot of carbohydrate chains coming off of it. So it's mostly carbohydrate with a small protein core. So just a little bit of that terminology there. And so these types of glycoproteins include collagen. So you've heard of that. So collagen, very, very abundant protein in our body, certainly very important for connecting up individual cells and tissues and providing structure to our skin, for example. Proteoglycans and fibronectin. So these are very important for forming this extracellular matrix. And then these extracellular matrix proteins are bind to receptor proteins that are anchored in the plasma membrane. And these are called integrins. And that name comes from the fact that these are proteins that are typically anchored. They're integral to the plasma membrane bilayer. So there's actually a nice little cartoon here that shows sort of an artist's rendition of what this looks like. So here's the lipid bilayer. So again, remember that on either side of the bilayer are these phosphate head groups. So these guys are hydrophilic, right? They're interacting with either the cytoplasm or the exterior of the cell. Both of those are aqueous environments. But inside, where these fatty acid chains are located, this interior is very hydrophobic. And so when we have integrin proteins, these proteins tend to be structured so that they prefer to reside in that hydrophobic environment inside of the membrane. So they're anchored there, and then they have various kinds of attachments on the surface. And for example, here's molecules that are called fibronectins. We have collagen, these long fibrous sorts of structures, and then here's proteoglycan that has these long carbohydrate chains coming off. Now these are very important for interactions that the cell makes with its neighbors. They're also really important for things like the immune system of our bodies, being able to figure out whether cells are self or non-self by looking at the kinds of external proteins and proteoglycans that are on the surface of the cell. So very important for the immune system to be able to recognize the extracellular matrix as something that looks like itself. Okay, and I want to talk a little bit about intracellular junctions. And as we said before, so neighboring cells in either tissues or organs or various organ systems, they have to be able to communicate with each other. And the way that cells communicate with each other is they exchange molecules. And these could be molecules of water, but they could also be salts. They could be other kinds of small molecules. And it turns out that in some cases, cells also exchange macromolecules like RNA molecules or proteins. All right? And so these kinds of junctions are very specialized structures, it turns out, that are set up to regulate that exchange of information. So cells really don't want to have just a highway going on between adjacent cells, because that's not a regulated, controlled process. Instead, they have very organized structures that allow the cells to control what's going back and forth between adjacent cells in a tissue or an organ. And I want you to know about 4 different kinds of intracellular junctions that we know about. Plasmodesmata we talked about before, so these are the types of structures that we see between plant cells. And then in animal cells, there are 3 different kinds of junctions that occur, and they have different structures, and they also play different roles in the physiology of the cell. And these are called tight junctions, desmosomes, and gap junctions. All right, so let's look a little bit at what those structures actually look like. So these are the plasmodesmata. These are channels that perforate the plant cell wall. It's a really beautiful picture right here that shows how this looks. So here's a kind of a slice through a piece of plant tissue where we can see one cell over here, and this is its interior. We see a second cell over here, and then you can see the cell walls, this sort of whitish area here and here. So those are fairly thick. And then what you can see is that here are the plasma membranes that are on the interior right here and right here. And these channels actually connect between the cytoplasm of the cell. So they form these structures. Anybody know what would these be made up of? These types of channels. Carbohydrate could be there, but also proteins, right? Would typically form that kind of structure. Let's go back. Right, so these are, and various experiments have shown that through these channels, course, water molecules and small kinds of molecules can pass, but also proteins and RNA molecules can pass between cells like this. So these channels are big enough to allow that to occur. And so that means that cells can actually exchange some genetic material, right? They can exchange RNA molecules. There's a process in cells that allows very small pieces of RNA molecule, very small RNA molecules to regulate the expression of proteins. And so we know that in plants, those very small RNA molecules that are regulating the expression of proteins can pass between individual cells, probably through these kinds of channels. And that is super important for allowing entire tissue in a plant to be consistent in the way that it's regulating expression of proteins, so that one cell isn't doing its own thing, where the neighboring cell is doing something completely different. Right? And then we have sort of a summary here that shows the way that these three different kinds of junctions in animal cells are put together. And this is a sort of a very cartoony kind of drawing that shows how these junctions work. And then these are actual micrographs that show what they actually look like. And so as I mentioned, so there's three different ways that these cells interact and can actually, in some cases, exchange molecules with adjacent cells. This here is called a tight junction. And as you can see, that name is very apropos because the cells are basically pressed right up against each other. The plasma membranes are very, very close. And these are, in fact, so tightly associated that water molecules really can't get through. So if you wonder why is our skin a good barrier to water? Water's not getting across our skin, certainly not easily. This is due to these kinds of tight junctions between adjacent skin cells that allow the cells to be, or at least this tissue formed by the cells to not be leaky to water and other kinds of small molecules. Okay, so that prevents leakage of extracellular fluid. Then over here, what you're seeing is what's called a desmosome. So these are really anchoring junctions. They're kind of like the rivets that hold cells together. So they help to fasten cells into a very strong kind of structure. And you can see just from looking at this that these are very highly organized structures that allow the cells to interact and to be very stably associated with each other. And then down here, what you can see are a series of gap junctions. So these are junctions that are really involved in communicating. And I sort of think that they're sort of the analogy in a way of the plasmodesma, right? They're basically a way that cells can exchange molecules between adjacent cytoplasm in adjacent cells is through these kinds of junctions. So they provide these cytoplasmic channels. And again, there's very nice evidence showing that in addition to allowing water and other kinds of small molecules to go between cells, that these can also allow larger molecules like proteins and some RNA molecules to go between cells. Okay, so let's now take a quick break. You can pull out your iClicker and let's just do a quick question, and then we'll talk about something fun. Okay, everybody got your clicker ready? Okay, so I'm going to start the timer and just take about 45 seconds to think about this. Vote your answer. Okay, so about 10 seconds. 5, 2, 1, stop. Okay, I have to get my iClicker software updated so I can actually show this on the screen, and I will do that. But what I can see here is that 89% of you voted for D, and that's correct, right? So microtubules are actually not involved in muscle contraction, right? What's responsible for muscle contraction? Yeah, microfilaments, right. A few people said C, so about 8% of you said C, and that's actually not the answer because we mentioned this briefly last time, but the nucleolus is the area inside of the nucleus where the ribosomal subunits are actually put together. So that's actually right. Okay, so that's where the ribosomal subunits are put together. And just in case anybody was confused last time, so the ribosome, and we'll talk about this more, but the ribosome is a 2-subunit machine that makes the proteins, right? And so the subunits, there's a big one, large one, and a small one. They're made separately, and they actually stay apart until they assemble on a messenger RNA, and then they come together. So that's a fundamental property of the ribosome. The subunits are made in the nucleolus, and then they get exported from the nucleus through these nuclear pores, where they interact with messenger RNAs in the cytoplasm and synthesize proteins. That's in eukaryotic cells. In bacteria, of course, they're not inside a nucleus, right? They're just in the cytosol. Okay, so that's great. And now I want to just tell you a little story that I think is really cool. It's not in the book, and it's really fun. So and it has to do with thinking about how scientists can use information that they have about the kinds of structures that we've been talking about and the ways that cells deal with how they deal with food, basically what they eat and what they make from these food molecules, sugar molecules, and how we can use that in an industrial setting. So we talked a little bit before about the fact that the plant cell wall, so we've seen these sort of micrographs, but they don't really show us the actual molecular structure of what's going on in that cell wall. And what's going on in there is that there are these layers and layers of cellulose. And so we talked briefly about the fact that cellulose molecules are these long polysaccharides. They're made up of, what's the subunit that makes them up? What sugar makes up cellulose? Glucose, yeah, right, glucose. So we've got glucose units, and so that's what you're seeing down here, are these individual glucose molecules. Of course, that's not the actual chemical structure, right? That's just a little sketch, right? But there's 6 carbon sugars, glucose. And these guys are hooked together. And in cellulose, they're actually hooked together in such a way that these are very, very stable, form very, very stable structures. They tend to be actually cross-linked through these hydrogen bonding interactions here. So they form these very highly networked molecules, very big macromolecules that are incredibly hard to digest because most enzymes can't really get in there and break down the linkages that connect up these individual sugars. Turns out some organisms have learned to do this. And one of these is called Neurospora crassa. So this is a type of fungus that's found. You can see it sometimes if you go out. You might see an orange fungus growing on dead tree branches, like you see right here in Hawaii, where sugarcane used to be grown, and where I grew up. You'd see sugarcane fields after the harvest, and these cane stalks, after being burned, would often have an orange fungus on them. We kids didn't know at the time that this was actually Neurospora. And what this fungus is doing is it's actually secreting enzymes that break down cellulose. They break it down into smaller units that this fungus can actually use for food. And so this is kind of a very schematic drawing showing these long chains that form these cellulose layers. And these are, again, very schematic drawings showing different kinds of enzymes that exist in Neurospora that are able to break down these chains. And when they get broken down, they typically end up in smaller molecules like this. They have either 2, 3, or 4 carbons in a chain. So they're called cellobiose, cellotriose, or cellopetrose. And so these can then be further broken down by Neurospora. But so that sounds great. And so you might think, okay, this could be cool. We could use this organism to break down cellulose and then turn it into ethanol, right? But it turns out that no, that doesn't really work well because Neurospora is an organism that requires oxygen to grow, so it's an obligate aerobe, and it also doesn't make ethanol very efficiently at all. But there is another kind of fungus that you probably know about that makes ethanol quite efficiently, and that's yeast. And so humans have known for several thousand years at least that you can use yeast to produce ethanol. But it doesn't work to grow yeast on cellulose because yeast does not have the kinds of enzymes that we find in fungi like Neurospora to either break cellulose down in the first place or to import those cellobiose and cellotriose type molecules into the cell. So they don't have the enzymes to break down those molecules, and they can't get them into the cells in the first place. And so this is just showing you that yeast grow very nicely on glucose and will make ethanol, but they don't grow on these guys. These are not food for yeast. So what's done industrially right now to make cellulosic ethanol, so if you read about that in the media, now you'll know how this is done industrially, is that what's done is to take these kinds of breakdown products from cellulose digestion and then treat them with an enzyme called beta-glucosidase that can be made in bulk but is very expensive to make. And so it can be made in a sort of industrial setting and then added to a mixture of these kinds of molecules. And when that happens, this enzyme will break these down into glucose molecules, individual monomers of these chains, and then those, of course, can get into yeast and make ethanol. But this is really expensive to do. So the laboratory of Jamie Kate here in MCB in chemistry had an idea about how to combine the advantages of Neurospora with yeast to make cellulosic ethanol. And the idea was to find the protein that makes this transporter in Neurospora that allows these cellobiose type molecules to get into the cells in the first place. And then they also found the gene that encodes this enzyme here, beta-glucosidase, that allows these cellobiose molecules to be degraded into glucose. And they engineered yeast to now encode both of these proteins, the transporter and the enzyme. And so when they did that, they now had a situation where yeast could actually grow on cellobiose, not directly, but by simply, let me just show you this whole thing. Yeah, so normally yeast would have to have glucose to survive and make ethanol, but when you have these engineered yeasts that have the transporter and the enzyme, these guys can actually grow directly on cellobiose. And now they have a very nice system using yeast to make cellulosic ethanol using this kind of pathway. And that's actually being licensed and is being used commercially now to make cellulosic ethanol. Very cool. See you guys Friday.